Assalamu alaikum. Second year. In today's class we are going to discuss the second lecture of the urinary system. Main objective of the today's lecture are to describe the histological feature of different part of nephron and then describe the juxtaglomerular apparatus. So renal tubule consists of the proximal convoluted tubule, the first segment of the renal tubule called proximal because it lies nearest to the tubule's origin from the woman's capsule, and convoluted because it has several bends in it. Then is the loop of Henle, the extension of the proximal tubules. Observe that loop of Henle consists of straight descending downward limb, a loop, and a straight ascending limb directed upwards. So distal convoluted tubule is the part of the tubule distal to the ascending limb of the loop of the Henle. It is extension of ascending limb of the Henle. And then is the collecting tubule, the straight not convoluted part of the renal tubule. Distal tubules of several nephrons join to form the single collecting tubules. So we will discuss all these structures one by one. The first is the proximal tubule. Main function of the proximal tubule is reabsorption of water, nutrients and solids. It is lined by the simple cuboidal epithelium, as you can see in the histological picture. The proximal convoluted tubules cells are simple cuboidal in nature. The tubule cells have several microvilli on their apical surfaces, which is forming the brush border, and these are active in endocytosis. Also the basal area, there are the striation in basal area due to numerous infoldings and plenty of the mitochondria present on the basal size. Terminal bar apparatus is present, and this terminal bar apparatus is formed by the tight junction and zonula adherens between the apical area of the adjacent cuboidal cell for the purpose of compartmentalization between the lumen of the proximal convoluted tubules and the intracellular compartment. Then is the plica, or the folds on the lateral surfaces of the cells, and extensive interdigitation of the basal processes of the adjacent cells. So these are the different features of the proximal convoluted tubules. You can see in this diagram. So simple cuboidal epithelium having brush border, having basal striation, having centrally located rounded nucleus and dark eosinophilia. The diagram, this is the diagram in the medullary area of the medullary ray. Here you can see large number of the proximal convoluted tubule. These are the proximal convoluted tubule. Again, this one is proximal convoluted tubule, and this one is the distal convoluted tubule. Again, this is the proximal convoluted tubules, and these are the collecting tubules. In this area you can see this is the area of the cortical labyrinth, and this area you can see many profiles of the proximal convoluted tubules because the proximal convoluted tubule is more convoluted, so that you can see many profiles in the cortical labyrinth area of the renal cortex. This is the area of the renal corpuscle, showing the vascular pole and the urinary pole of the renal corpuscle. Inside you can see the podocytes and the bunch of the capillaries, and this is the visceral layer. This is the distal convoluted tubule. Here you can see the proximal convoluted tubule, starting again another proximal convoluted tubule. So you can also appreciate that the lumen is irregular in case of the proximal convoluted tubule. This is the area of the medullary ray, and here you can see these are the descending thick segment of the descending thick proximal segments, and these are the ascending thick segments. So you can see in these areas the cells are more eosinophilic and resemble that of the proximal convoluted tubule. These cells have a larger size of the cells and decrease in number of the nuclei per profile as compared to the distal ascending segments. Next is the loop of the Henle, which is a U-shape and has descending and the ascending limbs. Thin descending segment has flattened epithelium, which is usually squamous in nature. It is permeable to water but not the solutes. So you can say these are the simple squamous epithelium present in the medullary loop. These function as the counter current multiplier system to determine osmolarity of the urine. The blood flow in the loop is counter to that in the medullary vasa recta. It extends from the corticomedullary junction to the medulla and consists of the descending limb, the thin segment, and ascending limb. Thin part is lined by simple squamous epithelium resting on the thin basal lamina. Squamous cells bulge into the lumen of the tubule. So this is the cross section of the medullary loop lined by the simple squamous epithelial cells. Next is the distal convoluted tubule. Straight part of the distal convoluted tubule is formed by low cuboidal cells without brush border. So in the slides of the proximal and distal convoluted tubule, you can easily differentiate between the two. The proximal convoluted tubules have a very well developed brush border because of large number of the microvilli, and in case of distal convoluted tubule there is no brush border. The diameter of the tubule gradually expands to about 35 microns, and convoluted part comes in contact with the glomerulus forming the macula densa. This is the segment of the distal convoluted tubule. Function is reabsorption, which is regulated by the ADH and aldosterone. It continues from the medullary loop and extends to collecting tubules within the cortex. These distal convoluted tubules are lined by the low cuboidal epithelial cells. You can easily appreciate that there is no brush border, although basal striations are also present in case of distal convoluted tubule, and these striations are because of the basal infolding and mitochondria, but number of mitochondria is less as compared to the proximal convoluted tubules. Its epithelial cells stain less osmophilic compared to those of the proximal convoluted tubule, and when you are appreciating, you are looking into the diagram of the cortical labyrinth, you can easily appreciate the two, the distal and the proximal, having the same outer diameter, but proximal convoluted tubule have less number of the nuclei as compared to distal convoluted tubules per profile. In this diagram you can see this is the renal corpuscle, this one is the vascular pole, this is the part of the distal convoluted tubule which is coming in contact with the vascular pole and forming the macula densa. Here you can see the podocytes are present. This is the Bowman's capsular space. Here this one is the urinary pole, and in this diagram you can differentiate between the distal convoluted tubules and the proximal convoluted tubule. This one is proximal, this one is proximal, this one is proximal, this one is proximal, and these three are the distal convoluted tubule. So in differentiating between the two, you can easily see they have the brush border, they have basal striation, the cells are larger in size, they are more eosinophilic, and number of nuclei per profile are less as compared to the distal convoluted tubule. The same diagram showing the proximal tubule. Here also the mesangial cells are present in the renal corpuscle. These are the smaller capillaries along with the podocytes present. This is the macula densa and the proximal convoluted tubule. This is again another diagram showing the distal convoluted tubule which is lined by the low cuboidal epithelial cells. These distal convoluted tubules resembles that of the collecting tubule, but you can easily differentiate between two because collecting tubules have larger outer diameter as compared to the distal convoluted tubule, and they have large number of the nuclei per profile in case of the collecting tubule as compared to distal convoluted tubule. Here in this diagram you can appreciate the thin tubules, the loop of the Henle lined by the flattened squamous epithelial cells, and these are the distal tubules, and this one is the proximal. These are the different parts of the nephron. The vascular pole, the efferent arteriole is entering and efferent arteriole is leaving, the Bowman's capsule, and then the urinary pole, the proximal convoluted tubule part, then the proximal straight segment, then the descending thin segment, loop of Henle, ascending thin segment, ascending thick segment, and then the proximal distal straight part and distal convoluted part, turning back towards its renal corpuscle of its origin, forming here the macula densa, and then attached to the collecting tubules by means of the arched connecting tubules. Here you can see the collecting tubules. These collecting tubules merge and form the larger collecting tubules called the collecting duct or duct of Bellini in the medullary area, and which enters into the minor calyx as the area cribrosa. This is the area showing this is the cortical labyrinth, and this one is the medullary ray. You can differentiate easily the renal corpuscles over here, and in medullary ray you can differentiate between the straight ascending and descending segments. Again, this is the area of the medullary ray, a cross section taken from the area of the medullary ray and surrounded by the renal corpuscles which are present in the cortical labyrinth. Again, medullary ray, the longitudinal section taking from the area of the medullary ray. Here you can appreciate this is the larger collecting tubules lined by the simple cuboidal epithelial cells. The another collecting tubule, here you can see another one, you can see another one, different collecting tubules, and these are the blood vessels forming the vasa recta. Again, another slide showing the area of the medullary ray. Here you can see the straight ascending and descending segment, and these are the area of the cortical labyrinth on each side of the medullary ray. Next is the juxtaglomerular apparatus. Juxtaglomerular apparatus is the microscopic structure in the kidney which regulates the function of the nephron. There are the three cellular components in the juxtaglomerular apparatus, which include the macula densa, the extraglomerular mesangial cells, and juxtaglomerular cells, also known as the granular cells. In this diagram, you can differentiate between all the components. This is the Bowman's capsule, and these are the macula densa forming the from the distal convoluted tubule forming the macula densa. In this diagram, so the juxtaglomerular apparatus contains the macula densa, lying directly adjacent to afferent and efferent arteriole and adjacent to some extraglomerular mesangial cells at vascular pole of the renal corpuscle is the terminal portion of the distal thick segment of nephron. It contains as part of its wall cells that are collectively referred to as macula densa because on light microscope cells of macula densa are distinctive in that they are narrower and usually taller than the distal tubule cells. Their nuclei appear crowded and they are superimposed over one another, thus forming the macula densa. The second component, which is the juxtaglomerular cells and the extraglomerular mesangial cells, which regulate the blood pressure cells. So juxtaglomerular cells are the modified smooth muscle cells of the tunica media of the afferent arteriole, and extraglomerular mesangial cells are just like the mesangial cells which are present in the mesangium, and these cells are present near the juxtaglomerular apparatus. So these three components, the macula densa, the juxtaglomerular cells and extraglomerular mesangial cells forming the JG apparatus. And these are the differences between the different parts of the nephron. The renal corpuscle is a capillary ball covered by podocyte and surrounded by simple squamous epithelial cells and the capsular space. The proximal convoluted tubules lined with the simple cuboidal epithelium and prominent brush border. Then the loop of Henle tubules that form loop. There are the thick and the thin ascending and descending portions. Most distal part of the loop often extends into the medulla. The thick limbs are lined with a simple squamous or cuboidal, and thin limbs are lined with the simple squamous epithelium. Distal convoluted tubules seem lined by the simple cuboidal epithelium with sparse or absent brush border. Cytoplasm of the cells tends to be paler paler than that of the proximal convoluted tuberous. So this slide was just a revision slide of this your lecture, and now this is the end of today's class. So we will meet inshallah in the next class. Till then, Allah Hafiz.